Hi, my name is Karolina. I'm a virology PhD student, and because I believe virology is full of fascinating stories, I decided to share some of them. When people think about viruses, one of the first things that comes to mind is that they are small. And to be honest, that's how I would describe them too. I mean, that's how we discovered viruses in the first place. They passed through filters that trapped bacteria, so we knew that this infectious substance had to be smaller than any known cell. So when scientists came across the very first giant virus, they naturally assumed it must be a brand new bacterium. In fact, this mystery microbe spent nearly a decade in a freezer before anyone realized it wasn't a bacterium at all. Instead, it was a virus. This discovery opened the door to an entirely new world of giant viruses, which had gone unnoticed simply because no one was looking for something so large in the virus realm. And it also raised an important question that still remains controversial. If some of these giant viruses could cause disease in humans. The first giant virus was discovered in 1992 during an investigation into a pneumonia outbreak in northern England. And the first person to see this mysterious microbe was Tim Robotham. He wasn't looking for a new virus. Instead, he was trying to confirm whether the pneumonia outbreak was caused by Legionella, a bacterium notorious for causing pneumonia, but very difficult to grow in the lab. So to do this, he perfected a technique called amoebal co-culture, which basically leverages amoebas as living incubators. Since Legionella naturally infects amoebas, Robotham was simply adding samples from the outbreak site into cultures of amoebas, letting any hidden microbes multiply inside. Afterward, he could more easily isolate whatever grew in the amoebas, Legionella, or, of course, some other microbe infecting the amoebas. After mixing samples from a cooling tower with amoebas, Robotham observed some bacteria, but he also stumbled upon something nobody recognized at the time. It was large enough to be seen under a light microscope, and it stained like a gram-positive bacterium. So it was mistakenly labeled as a novel bacterium and stored away. A few years later, the samples obtained from amoebal co-culture were transferred to a different laboratory, to Didier Raoult's laboratory in France. In his lab, scientists tried to study this assumed novel bacterium, but it was giving them a headache. The team tried to grow it on various bacterial media, but it just wouldn't grow. They also attempted to amplify its ribosomal DNA using universal bacterial primers, but they kept getting no results, and they assumed they must have been experiencing some technical problems with their assay. Finally, they turned to electron microscopy to observe the microbe at a higher resolution. But what they saw under the microscope most certainly did not look like a bacterium. Instead, they observed huge icosahedral particles, like some viruses have, but these particles were larger than any known virus. So after realizing it was not a bacterium at all, they named it mimivirus, which is short for mimicking microbe. So as the next step, the researchers naturally wanted to publish their exciting discovery. But at first, reviewers were actually very skeptical, and they wanted to see more proof that it was truly a virus. So the group sequenced mimivirus's genome, which was over 1 million base pairs long, so bigger than some bacterial genomes. And the findings were eventually published in the journal Science in 2003. Remarkably, By the time mimivirus was officially recognized as a giant virus, more than a decade passed since its first sighting. And much of the time it had spent tucked away, mislabeled in a freezer. The discovery of mimivirus certainly challenged our usual assumptions about viruses, particularly the idea that viruses are supposed to be small. In fact, mimivirus rivals and even surpasses some bacteria in both physical size and genome length. Its particles measures over 500 nanometers across, and it has multiple layers. At the center is the viral core, covered by two lipid membranes, followed by protein capsid that's covered in fibrils. And thanks to these fibrils, you can imagine mimivirus as a very hairy particle. These fibrils are also probably responsible for the gram-positive staining during the mimivirus initial discovery, leading to all that confusion. In addition to its size, mimivirus surprised researchers with the remarkable range of proteins it encodes. Because most viruses have only a handful of proteins. For instance, hepatitis B virus I work with encodes just 7. But mimivirus carries hundreds of genes for many different biological processes, such as DNA replication, transcription, DNA repair, as well as partial translation machinery, such as certain amino acid tRNA synthetases. And if you are not a biologist, I apologize for mentioning amino acid tRNA synthetases. But to me, what was the most fascinating feature of mimivirus is that majority of its genes, about 3/4, had no known equivalents in other species. These so-called orphan genes and proteins have unknown functions, and we cannot match them to our databases of known genes. Personally, I am excited to see how emerging artificial intelligence tools might help us reveal the functions of these mystery genes, given AI's growing ability to predict protein structures and interactions in a way we never imagined. So I think some answers might be just around the corner. So these were the 3 unexpected things about mimivirus: its size, the functions of the proteins it encodes, and its unique or orphan genes. But despite breaking these expectations, it still adheres to the key definitions of a virus. It is an intracellular parasite. It has a virion structure composed of nucleic acid in a protein capsid, and the virion does not multiply by division. And most importantly, even though mimivirus encodes some translation factors, it doesn't produce its own ribosomes or energy in the form of ATP. So it still has to hijack those vital functions from its host, like a true virus. The mimivirus's host is an amoeba, which is why it was discovered during the amoebal co-culture assay. And its replication was beautifully captured through electron microscopy. At first, its surface fibrils help mimivirus attach to the amoeba's outer membrane. And next comes the crucial steps. The amoeba actually eats the virus using a non-specific process called phagocytosis. Interestingly, amoebas usually consume bacteria of a certain size, generally at least 600 nanometers big. So one theory suggests that giant viruses like mimivirus evolved to be large enough to trigger this feeding response. So after being eaten, mimivirus sheds part of its outer layers by opening a special portal in its capsid called the Stargate, which reveals its inner membrane. Then the mimivirus escapes from phagosome into the amoeba's cytoplasm, where a viral replication factory is formed. The viral replication factory is basically created from both host and viral proteins, which the virus uses for replication. When researchers saw mimivirus's replication factory under the microscope for the first time, they actually mistook it for the amoeba nucleus because it was so large. The entire mimivirus replication cycle lasts about 8 to 12 hours, after which about 1,000 new virions are released from the cell, killing it in the process, before going on to infect the next unsuspecting amoeba. And soon after a mimivirus was described, scientists realized it wasn't the only giant virus out there. Since then, many other giant viruses were discovered, a lot of them also infecting amoebas. In fact, once researchers knew to look for large viruses, they started finding them everywhere, in oceans, lakes, wastewater, and in soil. Giant viruses share a few defining traits. They are typically over 350 nanometers in size, so they are big enough to be seen under a light microscope. They have genomes larger than 250 kilobases, and they often carry hundreds of protein-coding genes, with the largest known genomes exceeding 2 million base pairs. Like mimivirus, they are frequently harboring orphans, so the genes with no known functions or counterparts. And among the discovered giant viruses were some closely related new strands of mimivirus, but also more evolutionarily distant viruses with different hosts, shapes, and replication strategies. And naturally, by exploring this diverse class of viruses, the question arises: could any of them pose a threat to humans? Already shortly after mimivirus was discovered, researchers began asking whether it could infect humans and cause disease. After all, it was discovered during an investigation of a pneumonia outbreak. And the answer so far is a resolute maybe. Currently, about 20% of pneumonia cases in humans have an unknown cause. And there is some evidence that mimivirus might be responsible for some of these mysterious infections. One illustrative case comes from the French lab that was studying mimivirus. At the time, they followed typical rules for working with viruses which are not known to be pathogenic to humans. And in the lab worked also 38-year-old lab technician who was routinely tested for antibodies against the microorganisms he handled. And he had always tested negative for mimivirus. But then he suddenly developed a persistent cough lasting around 2 weeks, which was followed by fever, fatigue, and chest pain. When antibiotics failed, doctors took X-rays that suggested viral pneumonia. But standard viral tests came back negative. However, they found something unexpected. He had developed mimivirus antibodies, indicating a recent infection. Because of his previous tests, they knew that he didn't have these antibodies before. And now they detected antibodies against 23 different mimivirus proteins. Luckily, the story has a happy end, and the man eventually recovered. And the article didn't mention if he rather found a new job or continued to work in the lab. So this case makes a strong argument that mimivirus could be linked to pneumonia. But it is just a single case, and we should be very careful when evaluating all the available evidence. One approach researchers took was to look for antibodies against mimivirus in different populations. When searching for a pathogen responsible for a disease, you'd typically expect it to find it more frequently in sick individuals than in healthy ones. But with pneumonia, things get a little bit tricky because it has many possible causes. So even if mimivirus could contribute to it, you might not detect it in many pneumonia cases simply because those were the cases which were caused by something else. However, Even with this complication, several studies still found a higher prevalence of antimimivirus antibodies in pneumonia patients compared with healthy individuals. But the reported rates varied widely, ranging from as low as 0.04% to as high as 20% of patients testing positive. And on top of that, there are some concerns that there could be some cross-reactions in the antibody tests, which would lead to false positives. So this is making it harder to draw some firm conclusions. Researchers have also tried to look for viral genetic material in respiratory sample by using qPCR, and some have detected mimivirus DNA in patients with pneumonia. But again, detecting viral DNA alone doesn't prove active infection, because pieces of viral DNA might linger without any replication. Another angle of investigation is animal studies. I am aware of only one study investigating mimivirus pathology in animals, and in this experiment, mice were inoculated with mimivirus and control mice with inactivated mimivirus. And 9 out of the 12 mice given the live virus developed lung tissue damage, which was resembling pneumonia, and the researchers were able to re-isolate the virus from the lungs. But while this shows that mimivirus can damage lung tissue under experimental conditions, it doesn't confirm it as a definitive cause of pneumonia in humans. So overall, there is some evidence that mimivirus might occasionally cause pneumonia in humans. But I'm not fully convinced yet. What I find lacking is a proof that mimivirus can actually replicate in human cells. Specifically, I'd like to know which human cell types, if any, support the mimivirus infection and replication. A few studies have mentioned trying to infect human cells with mimivirus, but ultimately failed to detect productive infection. You might also come across papers stating that mimivirus can replicate in human blood cells, specifically in the PBMC cells. However, the same group that originally reported this later published a correction. Their initial findings came from an experiment where they inoculated PBMCs with mimivirus and then took extract of those cells and used them to infect amoebas, seeing a clear cytopathic effect in the amoebas. But upon closer inspection, they found no evidence of actual mimivirus replication in PBMCs themselves. There were no viral factories under the electron microscope and no detectable increase in viral DNA. And I really appreciate how they re-examined the data and published a follow-up study to correct the record, because that's science done right. As for whether mimivirus really causes pneumonia, we will see. Maybe the patients who tested positive had more amoebas in their lung, or they were co-infected with amoeba-related pathogen. One definitive test would be whether a hypothetical mimivirus vaccine could prevent pneumonia. But given how many different organisms can trigger pneumonia, that approach doesn't really make sense in practice. So for now, it remains an open question. Another mystery in giant virus research is where these viruses came from. And there are two main models. The first is reductive evolution. According to this theory, giant viruses might have started as ancestral cellular organisms. But over time, they adapted to parasitic lifestyle and they gradually lost genes they no longer needed and became increasingly reliant on their hosts. So the cells would gradually drop more and more functions, simplify, and eventually evolve into giant viruses. And the second model takes the opposite approach. It suggests evolution by expansion. This theory suggests that giant viruses might have started as small, simple viral entities, but gradually accumulated more genes over time. This could have happened through processes like horizontal gene transfer, or basically stealing genetic material, or gene duplications. For example, take mimivirus. When infecting amoebas, it could encounter other amoebal parasites, like bacteria or even other microorganisms. And along the way, it could swap some genes with them. And this would explain why mimivirus carries homologs, which are found in a wide range of species. One key point fueling the debate is the presence of translation-related genes, which are uncommon in most viruses. Some argue that these genes are leftover hints of a more complex cellular ancestor, supporting the reductive evolution idea. But other studies have shown that these translation-related genes have homologs across various species, suggesting they were probably stolen from different organisms, favoring the expansion model. Another fascinating mystery is the origin of the orphan genes, the completely novel genes with no known counterparts. Since these genes don't match anything in current databases, they can't simply be explained by typical gene transfers from known hosts. Some theories suggest that these genes could be a product of rapid sequence divergence, but others speculate that mimivirus and its relatives may have inherited these orphan genes from ancient, now extinct life forms. In other words, these genes could be relics from a distant past, lost in most modern cellular lineages, but preserved in the viral lineage, offering a potential glimpse into the evolution of life itself. Okay, up to this point I focused on mimivirus itself, but there's another twist. Mimivirus was the first virus known to have its own viral parasite. This came to light when a newly identified mimivirus strain was examined under an electron microscope. Amid its large replication factories, researchers spotted much smaller, 50 nanometers wide icosahedral particles, later named Sputnik. Essentially, Sputnik acts as a parasite of a parasite. And as an analogy to bacteriophages, it is called a virophage. Sputnik has a genome of roughly 20 kilobases with 21 predicted proteins, many of them novel orphan proteins. It attaches to the mimivirus to enter the amoeba, and it exploits the giant virus's replication machinery. On its own, Sputnik cannot replicate in amoebas. It needs the mimivirus replication factory. And the co-infection is actually beneficial for the amoeba host. Co-infection with Sputnik reduces the production of new mimivirus particles by up to 70%, and it weakens mimivirus ability to lyse amoebal cells. And future studies have since uncovered other virophages preying on various giant viruses. The discovery of virophages in mimivirus led to another intriguing but very controversial idea: the possibility that mimivirus might possess something resembling an immune system. It started in 2014 when a new virophage called zamilon was found to infect mimivirus. But not every strain of mimivirus was susceptible to the infection. Some strains showed an unexplained resistance. Researchers investigating why these strains resisted zamilon noticed a curious genetic feature. A 15 base pair segment of zamilon DNA was incorporated into the mimivirus genome, repeated 4 times. Disrupting the mimivirus gene containing these repeats made the virus suddenly vulnerable to the virophage. And For the biologists among you, I know there is already an alarm beeping in your mind, because this arrangement immediately calls to mind the CRISPR-Cas system in bacteria, where foreign DNA sequences are integrated into the bacterial genome and used as a sort of memory to defend against future invasions. The resistant mimivirus strains also had two nearby genes, neighboring the repeat, one encoding a helicase and the other one endonuclease. And knocking out either gene again rendered the virus sensitive to zamilon, suggesting that they might be involved in degrading the virophage's genetic material. The authors of the study, which was published in the journal Nature, dubbed the system Mimivire and proposed that it might act similarly to CRISPR. But the Mimivire hypothesis drew immediate scrutiny. Some scientists criticized the comparison between the Mimivire system and CRISPR, arguing that these two are structurally different. Unlike CRISPR, the repeats in Mimivire are not regularly interspaced, nor are they flanked by sequences that would help to distinguish between the host DNA and the invading DNA. And this key difference raises doubts whether the Mimivire system functions in the same way as CRISPR, or if it represents a completely distinct antiviral defense mechanism. Another suggested theory speculates that the zamilon-derived segment, which lies in the protein coding region, might generate a peptide that could be disrupting the virophage replication by some other mechanism, for example by competition for binding to some replication factor. But so far there is no experimental evidence for this hypothesis, and how Mimivire works remains a mystery. And before I wrap up this episode, there is one important issue I feel that should be mentioned. And that's the controversies and ethical concerns surrounding some of the researchers behind many of the discoveries I've discussed. The main figure at the center of this issue is Didier Raoult, but other scientists involved in giant virus research have also been implicated in some of these concerns. Now, when I was reading the papers on giant viruses, I didn't find anything problematic in those specific studies. However, many of Raoult's other works have been retracted in recent years, and more than 200 of his research papers have been questioned or flagged with concerns. From what I read, the most common issue includes conducting studies without proper approval from ethic committees or obtaining approvals that didn't actually cover the research they carried out. In some cases, there have also been reports of problems with published data, such as image duplications. And if you're interested in this topic, it has been extensively covered by sources like Retraction Watch, which tracks scientific misconduct and retractions. As for the papers I used as sources for this episode, I checked them on Papier, which is a platform where researchers discuss potential issues in published studies, and I didn't find any flagged concerns related to the articles I referenced. And well, that makes for ending the episode on a slightly depressing note. So I guess the take-home message is giant viruses are awesome and people are a bit less awesome. And with that wonderful remark, I want to thank you for listening to this episode, which makes me very happy. And I also want to thank everyone who has rated or recommended the podcast. Your support really means a lot to me and I appreciate it. The podcast is also available in Czech, hopefully with better grammar and pronunciation, and you can find the episode sources in the description. If you'd like to be notified about new episodes, you can follow the podcast in your app or on Instagram. And once again, thank you for listening.